So hello everybody, today I'm going to be talking about a project I've been working on where I've been trying to do saturation variant interpretation using CRISPR prime editing. So to give a bit of context for what I've been working on, we all know that the human genome was sequenced for the first time in about 2001, but since then the cost of sequencing a genome has fallen precipitously, such that now sequencing a whole genome has become rather routine. As we aggregate all the data from these genomes, such as what's being done by the Nomad team, we found a staggering amount of variation across the human population. So what I would argue is that the new Human Genome Project is trying to understand what all of this variation does. And I would argue that we're not doing the best job of that right now. So if we look at all of these documented variants, we find that less than 1% of them have an associated clinical interpretation. And so there's many ways you can go about linking a given variant to a clinical or functional interpretation. What I'm going to be talking about today is something called multiplexed saturation genome editing. And what that entails is delivering a Cas9 enzyme alongside a library of donor templates, each one encoding each of the possible nucleotide substitutions across your targeted region. You deliver that to a pool of cells. Hopefully each cell gets one of the possible mutations, and then you have to perform some functional assay that will help you understand what the functional significance of each of those variants are. So if we were to do that in this example here, we can say that our functional assay results in death of any cell containing a pathogenic variant. So if we delivered our CRISPR library to the cells here, each one getting one of the possible mutations, we would then sequence that population, perform the functional assay, and then sequence that population again. And what you would notice in this example here is that there's a complete loss of the green cells and a reduction in the red cells, whereas the blue and yellow cells stay about the same. And from that, we might infer that the red and the green cells carry pathogenic variants, whereas the blue and yellow variants are benign. And so this approach has been used to great effect. Jay Shenduri's group published the interpretation of about 3,000 different variants in BRCA1 using this method. When I began working on this project, however, there were two things that I wanted to address. The first is that with most genome editing technologies, you get really low product purity. And what I mean by that is if you have a diploid cell line, you're much more likely to find a cell where each allele has two different mutations on it than you are to find editing outcomes where both alleles have the exact same editing outcome. As a result of this, it becomes really difficult to disentangle the effect of any given mutation. The way the Shenduri group got around this was by using the HAP1 cell line. So HAP1 cells are a near haploid human cell line, which makes them great for looking at mutations in isolation. The downsides, however, is that HDR is prohibitively low. Their ploidy is highly unstable, so it actually takes quite a bit of work to maintain these cells as haploid in culture. And then obviously, given their haploid nature, there's some questions about their physiological relevance. So what I wanted to do was develop a strategy where we could get the benefits of the HAP1 cells while sidestepping some of those downsides. And the idea I had was to generate haploidized gene loci. And what I mean by this is that you take your diploid cell and you just generate an allele-specific deletion on one of the two alleles so that you would lose a fragment on one allele while the other one remains intact. And so what this looks like in practice, this is an example using the NPC1 gene in HEK293T cells. And if you look at the sequencing here, you can actually tell that the locus here is triploid because the A nucleotide is represented in about 1/3 compared to the G nucleotide in both instances. So what we wanted to do was develop an allele-specific CRISPR strategy where we would just get cleavage on the two alleles containing the G nucleotide in each cases, and no cleavage on the allele containing the A nucleotide, which would generate deletions on those two alleles while leaving the other one intact. And so after we do this, this is what you look like. This is what you find now. So the sequencing traces here appear to be homozygous for that more minor A nucleotide in each cases, indicating that those other two alleles have been removed. And so what I would argue is that this haploidization strategy generalizes saturation genome editing. So these haploidized cells are great in that you can effectively model mutations in isolation, and they can be translated to any given cell type, including non-cancer stably diploid cells such as RPE1. So ultimately, cell line selection can be made based on phenotype or physiology rather than a strict reliance on this HAP1 cell type. So with these haploidized cells, I started to work on a project surrounding a disease called Niemann-Pick disease type C. So Niemann-Pick disease type C is a lysosomal storage disorder caused by autosomal recessive mutations in a gene called NPC1. So this disease is fatal by about ages 15 to 25, and the hallmark symptoms include a global neurodegeneration and enlargements of the spleen and liver. And all of these symptoms result of an inability to transport cholesterol throughout the cells. So NPC1 is a lysosomal protein that will move cholesterol from the lysosome to other cellular compartments. And what makes NPC1 a good candidate for saturation genome editing is that the genotype-phenotype relationship is really poorly understood. So to date, more than 400 disease-causing mutations have been identified. Most patients present as compound heterozygotes, often harboring one mutation that's entirely unique to their family. And so as a result of this, it makes it very difficult to understand the impact of any given mutation, which has left our ability to predict the phenotypic outcomes of any mutation as really poor. And obviously, this is going to impede treatment administration and genetic counseling efforts. So I, again, am using this haploidized cell line. And what I'm showing you that's new here is a Western blot, which shows that in the haploidized cell line, you have about 1/3 the protein expression as the parental wild-type cell line. And so this indicates that this cell line is functionally haploid. So it's only expressing NPC1 from that one remaining allele. And so instead of using the HDR-based strategy that I outlined previously, I was interested in using this new CRISPR prime editing technology. And so very briefly, the way this works is you have a Cas9 nickase enzyme, which is fused to a reverse transcriptase. And the other difference is that it uses something called a prime editing guide RNA. And the difference between a prime editing guide RNA and a normal guide RNA is that a prime editing guide RNA, or pegRNA, has an elongated 3' extension. And on that 3' extension is actually where you encode your edit of interest. So what happens is the Cas9 comes, it binds, it nicks the PAM-containing strand, which then hybridizes with that elongated 3' extension. The reverse transcriptase then reverse transcribes whatever you've encoded in that 3' extension into the genome. Through a series of repair steps later, you end up getting your edited product. And so what I was interested in was how multiplexable this prime editing could be. So the good thing about prime editing is that you can use the same pegRNA architecture to introduce mutations over a relatively wide window. So here you can see a C to T transition right at that pegRNA nick site, and you can tell just by looking at the Sanger sequencing read that the editing efficiency is quite high. But using that exact same pegRNA architecture and just swapping the nucleotide within the 3' extension there, you can encode an edit, in this example here, 31 base pairs away, another C to T transition. And what you can appreciate is that the editing efficiency does not seem to drop precipitously as we go farther and farther away from that nick site. So I designed a small pilot experiment where I designed a library of pegRNAs, which would encompass every possible nucleotide substitution within a 16 base pair window of NPC1. And what I wanted to understand was whether you could effectuate saturation genome editing using this strategy. And so I performed next generation sequencing on the edited population, and what I found is that yes, indeed, you can get saturation genome editing. So each base, you can find its single nucleotide substitution to every other base. And there's two other things worth noting here. First is if you look at that top row there, that's the percentage of reads that map to wild type. And you can see that 42% of sequencing reads were mapped to the unmodified allele, which indicates that 58% of reads were modified, so you get really high editing efficiency with prime editing. And another thing, you can tell that each of these reads has a C to A mutation alongside the targeted mutation. So that mutation disrupts the PAM site from Cas9 rebinding and re-editing. In our initial experiments, we thought that that might be necessary to ensure coverage of the entire library, effectively so that once you get one edit, you don't have the Cas9 come, bind again, and re-edit that locus. In our subsequent experiments, actually, we found that that's not necessary, so you can actually just introduce your edit of interest with no PAM modifying mutation, which makes this unique to HDR-based approaches, which necessitate that PAM modifying mutation in addition to your mutation of interest. So now I had my cell model and my method for editing. The next I needed was that functional assay to delineate a functional from non-functional variant. And so what I did was develop an assay based on the observation that NPC1 deficient cells have an expanded lysosomal compartment. So if you stain NPC1 deficient cells with the fluorescent dye called lysotracker, which is just a dye that will accumulate within the lysosomes of cells, you will find that the NPC1 deficient cells, which are on the right here, are much brighter than the wild type's equivalent. The great thing about this is that it seemed to translate well to a FACS-based assay. So by performing fluorescence-activated cell sorting on these cells, you can tell there's a pretty distinct separation between the wild type and NPC1 null cells. What I was further interested in, though, before continuing, was understanding the sensitivity of this assay. So basically, NPC1 is characterized by a very heterogeneous disease presentation. So some patients will develop symptoms at birth, and their disease will progress very quickly, whereas other patients might not start to develop symptoms until they're 8 to 15 years old, and their disease might progress very slowly. And so a little bit of this can be attributed to mutations. So there are mutations that are associated with sort of later onset, less severe disease, and mutations that are associated with very typical severe disease onset. And so I'm using this prime editing system. I made a few of those models. The first is the NPC1 P1007A, and so that's typically associated with a very late and mild disease progression. And the other one is NPC1 I1061T, which is associated with very severe and early disease onset. So I isolated each of these models, and you can tell just by looking at the Western blot here that these mutations clearly have these mutations have very different impact on protein expression. So P1007A looks relatively indistinguishable from wild type, whereas NPC1 I1061T has reduced protein expression, and it runs at a lower molecular weight than wild type equivalent. And so by staining these populations, I performed the lysotracker staining and then the FACS sorting of each of these clonal populations. And encouragingly, what I found was that you can distinguish between, not only we could tell the difference between a wild type cell and this NPC1 P1007A, which is a typically very mild disease-causing variant. So that's great. We are sensitive enough to distinguish between even mild variants. But what's great too is that the NPC1 P1007A looks even distinguishable from NPC1 I1061T. So not only might we be able to distinguish between a disease-causing and a functional variant, we might be able to start discerning relative functionality of different variants based on this assay. So what we did was develop this FACS-based pooled assay. So what we would do is we would design a library of pegRNAs. So we would take one target site, and these would range from about 13 nucleotides to about 50 nucleotides. So it would be about 40 mutations to 150 mutations simultaneously. We would deliver one of those libraries to a pool of cells. Each cell would get one of the possible mutations, and then we'd stain the cells and run them through the FACS sorter. We then gated the population into a low and high fluorescence population, and this was based on what the staining, what the lysotracker's signal distribution of the wild type population looked like. Basically, we split that distribution right down the middle, with the bottom 50% of wild type control cells corresponding to a low fluorescent population, and the top 50% of control cells corresponding to a high fluorescent population. So the assumption would be, once we sort these out, extract the DNA and sequence the target locus, if we calculate the log-fold change between those two populations, so how each mutation is distributed across those two populations, we would expect a benign variant to act like wild type, so you'd find it about equally in both the low and high population. But pathogenic variants, since we know those accumulate more lysotracker and are brighter, we would expect those to be disproportionately skewed towards the high population. And so by just sequencing these two populations and then quantifying the log-fold change across these two populations, we were able to develop a function score for each of these variants. And so that's what you can see on the left here, is just a histogram distribution of those scores. And what was encouraging at first is that a lot of our assumptions that we would make about a saturation genome editing experiment appear to be true. and that is that synonymous mutations all seem to cluster together, a function score of about 1, and nonsense mutations all cluster together but apart from the synonymous mutations with a functional score of about 0. So what's great is you can actually use these function scores to classify variants. So we've developed an unsupervised clustering algorithm which tries to identify the wild-type cluster and then classifies everything with a score lower than that as deleterious. And so we've applied this strategy over about 16 different targeting experiments, so 16 different libraries. And in those 16 different experiments, we classified about 978 different NPC1 variants. And so we calculated a function score for each of those and then performed this unsupervised clustering. And what you can see on the right is the result of that clustering. And what sticks out is that NPC1 is clearly very sensitive to genetic perturbation. The vast majority of missense mutations are deleterious. What's great is that, again, you see that the synonymous mutations are almost invariably functional, whereas the non-synonymous mutations are invariably deleterious. And so, as I mentioned before, we were a bit interested in understanding not only can we classify this as deleterious or functional, we're interested in understanding if this function score actually means something about disease pathogenicity. So, as you can tell in the histogram here, the missense mutations have quite a wide spread. So, if a missense mutation has a score of about 0.5, is that meaningfully different from a missense mutation that has a score of about 0? And so to start addressing this, we isolated about 60 different clonal cell models from our different screens. And so what you can appreciate is that you can get pretty distinct impacts on protein expression and trafficking depending on the mutation that you're looking at. And so this is pretty well established. So NPC1 undergoes pretty extensive post-translational modification as it traffics towards the lysosome. And so this is reflected in the Western blotting you can see. So some variants run as just a single lower molecular weight band. Those are variants that are unlikely to have been trafficked successfully whatsoever. And then you have some that run as a normal molecular weight, but with a reduced protein expression. And then you have some that are a bit in between. And so we came up with 4 different categories based on the Western blotting from about 60 of these different clones. And what we found was that the category that we grouped them in seemed to correspond quite well with the function score. So variants that ran just as a lower molecular weight band, indicating that they're unlikely to be successfully trafficked to the lysosome, tended to have scores that were really similar to the average nonsense score, which makes sense. If the protein's not making it to the lysosome, then it's unlikely to be doing anything functionally productive. And comparing that to a variant that's in protein status subgroup 1, which is a variant with expression comparable to wild type, you find that the function score is actually quite a bit higher, which would make sense. And so to follow this up, we're interested in understanding whether we can draw any correlations between the function scores we've derived compared to known clinical outcomes of patients with these mutations. So not only can we classify something as benign or deleterious, we might be able to have some insight into what disease progression might look like just based on this functional score. And to take this one step further, we wanted to confirm that the interpretations that we were making were not unique to the HEK293T cell line. So we actually haploidized the RPE1 cell line, which is a stably diploid cell line. And by chance, it shared the same haplotype as HEK293Ts. So we were able to use the same guides that we used to haploidize NPC1 and HEK293T cells to haploidize this RPE1 cell line. At the same time, we made a knockout cell line to confirm that the phenotype would be the same. And what we found, as you can see here, is that the NPC1 knockout RPE1 cells clearly accumulate a lot more lysotracker than the wild-type counterparts. Another question was how efficiently we were going to get prime editing to work in these cell lines. We took 2 of our verified libraries from the HEK293T cells, delivered those to these RPE cells. What you can see is that you get very robust editing efficiencies. So we delivered these libraries, did the exact same experimental protocol we would do with the HEK293T cells, sorted the population, sequenced them, derived the function score, and classified them. And what we were very encouraged to see was that there was a very high correlation between the function score made in HEK293T cells, which is along the x-axis here, and the same function score that we would derive from the RPE1 assays, which is on the y-axis here. And so this seems to suggest that the function scores we've derived are highly reproducible across distinct cell lines. And so to summarize, we've classified 978 different variants across 16 different pegRNA target sites. The target sites varied from 13 to 43 nucleotides. One thing worth noting is that we did pre-screen each pegRNA in advance for activity. And so, and in addition, where compared, our scores are highly reproducible across the 2 different cell lines assayed, and our scores seem to reflect unique molecular defects within the NPC1 gene. And so with that, I'm going to leave off. If you have any questions, you can send me an email or find me on Twitter. If you scan the QR code here, you can read more, which is all detailed in the paper we've put on bioRxiv. So thanks a lot for your time.